Okay, thanks. So a fair amount to talk about. Give you a little background on the company is primarily what we do is clone antibodies as therapeutic candidates. And we clone them directly from human B cells. So the platform that we developed in order to do that is pretty powerful. And the result of that is that I think it's useful for the kinds of issues that we're talking about at this conference. So just a little background is the technology is mature at this point. We've applied it to half a dozen different projects. And in each of those cases, the antibodies that we generate are qualitatively better by quantitative objective measures like affinity and specificity compared to our closest competitors. So it's a mature technology. It works. The people involved are all veterans of biotech and pharma. So hopefully we have a little bit idea of what we're doing. So actually put this slide in and was sort of expecting that a lot of this material would have already been covered by other speakers, but we didn't really get into a lot of this in the earlier talks. So let's spend a few minutes on the question of what the problem is with anti-drug antibodies. So even when you're injecting drugs that are by sequence the same as the natural human protein, there's still the opportunity for unnatural route of presentation at unnatural levels and slight modifications during manufacturing to break immune tolerance, and you end up generating anti-drug antibodies. Now, for industry, this is a big problem because cross-reaction to normal human antigens poses a significant safety issue, and that's both off-target reactivity as well as antibody neutralization of the endogenous version of the protein, or that the anti-drug antibodies can mediate cross-linking of the drug, and that would make it trigger unwanted results. Likewise, the level of anti-drug antibodies, if that varies among different people, that can be a sponge that affects the PK/PD results. So you think you're giving the same dose to everybody, but if they have different levels of ADAs, it's hard to interpret your dose response results. And then these days, as the industry matures and generic versions of biologics are coming along, they may differ significantly in their induction of anti-drug antibodies. And so now you get into a chicken and the egg problem. If it's a biosimilar, then you have to show that it has the same ADA profile. If it has a better ADA profile, then it's no longer a biosimilar, and now you would have to do separate efficacy trials. So it's a mixed bag, but probably the FDA is going to be very interested in this going forward. So the guidance that the agency has given so far is that they want drug sponsors to address the issue. And they're picking carefully their targets on this. So Halozyme has been in the gun sights initially because their drug is a modifier of other drugs, and so it's not essential. And so the FDA has been really strict with them to prove that the ADA responses to hyaluronidase are not a problem. And that's not an easy thing to do, to prove a negative, that it's not a problem. And you can't really do it in animals because the animal models aren't predictive, is that that means there's a high premium on generating ADA data in man. So that's the problem, is that ADAs certainly exist. The question is, to what extent are they a problem? Are most of them benign? Are some of them a problem? And how do you tell the difference? So Trellis' solution to the problem is we've got this great technology for cloning human antibodies. So let's just clone out the ADAs, is that trying to understand what's going on at the serum level. It's really difficult to tell the difference between an antibody of moderate affinity that's at high abundance compared to one that's of high affinity that's at low abundance. And trying to tease those out from each other is really difficult at the serum level. Cloning them should make it all a lot easier. So if the idea is to understand which of them are a problem and which of them are benign, what we need to do is be able to clone a comprehensive suite of antibodies, that is, ones that march all the way around the surface of the protein. And there are side benefits to generating a comprehensive suite, is that if you have antibodies that bind to different regions of the protein, then it makes it much easier to construct sandwich assays, which typically have higher sensitivity, higher specificity, so that's useful. And then if we have the ability to look at the repertoire with cloned antibodies, now you can do competition binding assays. And so those are cheap and easy. You can run large numbers of them in order to assess population frequency of ADAs to different epitopes, track the evolution over time, and so forth. And then from a safety point of view, if you've got cloned ADAs, then you can use those to measure off-target reactivity in the usual way of staining a panel of human tissues. If you find some binding to a particular tissue, then you can also say, well, okay, let's investigate whether that leads to any toxic sequelae. So that's the program, is we want to clone a comprehensive suite of antibodies. So let me take just a couple of slides to summarize about 5 years of work, about $40 million worth of venture capital, and it all sounds simple in retrospect, but it wasn't so easy to get all of this working. So I think most of this audience is familiar with a Tcell Elispot assay. So think of it as an Elispot assay for B cells with a couple of additional features to it. So the notion here is that we'll take PBMCs out, we'll plate the B cells out on a surface, and over the course of a few hours, each B cell secretes a few femtograms of antibody that we capture because the surface they're sitting on has been coated with a capture reagent for IgG. And so what you see in the top panel in the middle, the fuzzy green spots there, that's about 200 or 300 B cells in one well of a 96 well plate. And each of those fuzzy green spots is the secreted IgG from a single B cell. So obviously we're not so much interested in counting how many IgG secreting cells there are, we're interested in antigen specificity. And it turns out when you're in a really microscopic assay environment like that, in order to get a signal out of that, you need a really bright label. So the bright label that we use are fluorescent microspheres that are filled with fluorescent labels. And actually we came up with the idea about the same time that Luminex and Illumina came up with the idea of combinatorial coloring. So the beads actually have two different dyes in them, a red dye and a green dye, and the ratio of those determines what the bead type is. For illustration purposes, it's just easier to give them different colors because your eye isn't very good at seeing small differences in ratios. So on the bottom there, now we see that within that well there are 3 cells that react with the orange-colored beads that are coated with antigen E. And it turns out that when you have multiple different criteria that you're defining as what's a B cell of interest, the frequency is pretty low, on the order of 1 in 100,000 to 1 in a few 100,000 B cells. And anyone who's ever tried to look for things that rare by flow cytometry, it's really, really hard because you're not sure when you've got a false positive. So the fact that we see replicates in the same well, which are daughter cells of the original cell, it makes it really a lot easier to pull out rare cells. So that's the basic concept. Let me give you one more slide with a little bit more of the details of the mechanics of how it works. So typically we'll start with 10 cc's of blood, and out of that we get about 250,000 B cells that we can kick into a proliferative mode that's secreting IgG that lasts for about 10 days. And then the cells start to die, or they stop producing message for the IgG. So we take those B cells, we plate them out at about 300 cells per well in 96 well plates, and we grow them for about 4 days. So during that time, there's several rounds of replication. Now each of the original B cells is represented by multiple daughter cells. take an aliquot of that, put it in a new plate, spin them down to the surface, let them secrete antibody for about 3 hours, wash out the cells, and now we have a very clean assay environment. And then we stain those with the different antigens on the different colored beads. So now the colorful picture over on the top panel there, that's the high magnification view of the footprint of a single B cell. And the little spots of light there are the individual fluorescent beads. So computerized microscope goes through and counts up how many red beads, green beads, blue beads, yellow beads, and basically boils down gigabytes of raw image data down to a few kilobytes of informative data in real time. As I said, typically it'll be one well out of every 2 or 3 microplates that meets the criteria of interest. So then we go back to that well, which has continued to grow in the meantime, and we take that one well and we do limiting dilution cloning out into 384 well plates. This time do a quick and dirty assay just to find the wells that are reactive with the antigen, and then take those single cells and do PCR, single cell cloning of the heavy and light chain. So the hardest part of all of this was to get the whole thing compressed into the 10 days that's basically the lifetime of the B cells once you get them outside the body. So just a little B cell fun for the aficionados here is by the time we assay the cells, we've kicked about 70% of the resting memory B cell population into a proliferative mode that's secreting antibodies so that we can assay it. This is actually my favorite slide of the entire project, is that multiplexing turns out to be really useful, is that it allows us to incorporate secondary screening criteria into the primary screen. And that means that you have to clone many fewer B cells at the end of the day to find ones that have the property that you like. Now, multiplexing is only useful if the assay is reproducible, because what you're looking for are ratios in the binding in the different data channels. And so this is looking at 3 sibling cells, 3 descendants of a single primary cell, stained with 4 different antigens from our influenza discovery project. And when you realize that what we're looking at here is a single cell assay with a few femtograms of antibody, and we're probing it with 4 different probes, each of which have a dynamic range of at least 10-fold. And the reproducibility of the assay is just absolutely staggering for an assay that's this miniaturized. And the reason for it is that there's no liquid handling steps. Most miniaturization assays, where the noise comes in, is when you're trying to move very small amounts of liquid from one place to another. So supernatant assays have nowhere near this level of precision in the assay. It's because the antibody is stuck, and then we bring the assay to the antibody rather than the antibody to the assay, that we get this high reproducibility. So this is just to assure you that, yes, the technology really does work. We've done it on 3 different virus projects, for which the resulting antibodies, we've been able to characterize them compared to our closest competitor. And in each case, we're able to look at many more clones at greater depth of characterization. And so not surprisingly, we end up with better antibodies. Now, just by luck of the draw, on the 3 different projects, our closest competitor on each project used a different one of the 3 major technologies for isolating antibodies. So either supernatant assays or flow cytometry or phage display. And for technical reasons that either involve much higher throughput or multiplexing, one or the other or a combination of those is the reason that this technology actually should generate better antibodies than the other ones, and in fact that's the way it's been working out. Okay, so applied to anti-drug antibodies, one of the issues that is real important in this area is the sensitivity of the assays. So most of the anti-drug antibodies that are likely to be a problem are going to be present at a low level. If they were at a high level, probably the drug never would have made it into the clinic. It would have been obvious preclinically, or very early in phase 1, it would have been ruled out. So the kind of issues that the FDA is worried about, that the people in industry are worried about, they're inevitably going to be relatively rare antibodies. And so the question is, how do you get a really sensitive assay for them? So just comparing Amgen's published sensitivity of their ELISA assay for EPO, is that if we just run this bead-based assay just on looking at a fixed unit of area and counting how many beads, it's about 100-fold higher sensitivity. And then the actual area that we're looking at in the footprint of a single cell is probably another 5-fold or so increase in sensitivity. So we've got a really, really sensitive assay. And the implication of having a sensitive assay is that you can see things that are below the level of detection by conventional assays. So this is just an example. We did an experiment with a friendly company down the road, and they had made a bunch of hybridoma libraries, and they were screening them by standard supernatant ELISA. So they would grow the cells up, and they would wait a couple of weeks, and then they would measure the supernatant. And then we took those cells at the early stage before they grow them up, and we just did a cell spot assay on them. And it turns out that about 30% of the wells that scored negative by the ELISA assay actually do contain antibody-producing cells that recognize the antigen. They're just not producing enough of it to be detected by the ELISA assay. So if you can find more things, then that's going to be a good thing. So now we have to address the question of, well, okay, you've got sensitivity to beat the band. How much sensitivity do you really need? And this is a topic that I think deserves a fair amount of discussion in a group like this. Let me give you some initial thoughts on it, and would be curious to know what other people's thinking on it is. So the way I've been thinking about it is that ADAs are not going to be clinically relevant if they're present at significantly below their affinity. So they're just not going to be able to bind very much if they're present at a concentration that's below their KD. So calculate it out for a typical KD of 1 nanomolar on 150,000 Daltons for an antibody. That means it needs to be present at about 150 nanograms per mil. And a typical antibody level in blood is, let's say, 10 to 15 mg per mil of all IgGs. So that says that the relevant antibodies should be present at more than about 1 part in 100,000. And that's a nice number because the belief is that when you generate a plasma cell, you generate a memory cell as an asymmetric division. And the memory cell compartment should be roughly proportional to the level of ADAs. So that says that if we're going to look for ADA-producing B cells in the memory compartment, we need to have a resolution on the order of 1 in 100,000. And we've got a resolution of about 1 in 250,000 from each individual donor. And it only takes a few days to do a survey of a donor, so we can do 10 or 15, 20 different donors. So that says that this assay should have sufficient resolution to pick up any of the ADA-producing B cells that would be physiologically relevant. If they're more rare than that, they're probably not important. Okay, let me give you an actual example of what the B-cell repertoire looks like when you use this kind of technology. So this comes from our RSV therapeutic antibody discovery. And what you're looking at across the bottom is 24 different blood donors, and the stacked vertical bars, the different colors are different specificities. And in that particular example, the combination of features that we are interested in is the one that's marked in red. And you can see that only about a quarter of the donors are making any level of antibody whatsoever that recognizes the phenotype marked in red. And even the ones that are making it, the frequency is a few parts per 100,000 B cells. And it's actually the fifth one over there with just a few clones per 100,000 that actually delivered our therapeutic candidate. So on the top panel, in terms of positioning this within the ADA context, is that we're interested in having adequate sensitivity to find rare things. And so that's the top row, is that, yes, we can find rare things. But equally important is that if you don't find it, how reliable is that negative result? And the answer is that in this assay, we get multiple chances to see the antibody producing cell if it's really there. So if it's not there, a false negative rate should be pretty low. If it's not there, if we don't see it, it's because it's really not there. Won't go through the details of this. It's just to make the point that obviously we can sample the repertoire at different points of time, so you can look at the evolution of the response. And now I want to spend a little bit more time on this question of how do you get a comprehensive suite of antibodies that would march all the way around the surface of the protein. So the simplistic way of doing it is just to brute force it. Let's clone a large number of antibodies, and then we'll test them pairwise against each other for competitive binding. So that's pretty easy to do these days with small amounts of antibody that you can get from a quick transient expression of the cloned antibody. And we do it with a Forte Bio BioCore, and any biosensor would work fine. So on the left side of the lower half of the slide there, two different antibodies that both bind the hemagglutinin from H1 strain of flu. And they bind at different sites so that you can get additive signal on the biosensor. On the right-hand side, the two antibodies compete for binding at the same site. So once you've bound one of them, then add in the next one, you can't get any increase in signal. So it's a pretty quick and easy assay. You can look at large numbers of different pairwise combinations. And you can take this large suite of antibodies and condense them into competitive binding classes. And then the idea is you just continue screening until you get to an asymptotic point where you keep screening, you keep cloning new antibodies, and you don't find any that represent a different competitive binding class than the one you already have. And at that point, you say you're done and pick one representative from each of the competitive binding classes, and that's your comprehensive suite. So that's a labor-intensive way of doing it. We think there's a shortcut that should work for most of the proteins of interest. So what's needed for the shortcut is we have to have a high-resolution structure of the protein. And these days, either by homology modeling or X-ray crystallography and other techniques have become so powerful that most drugs that are heading to the clinic probably do have an adequately high-resolution structure. And the game here is that we want to predict the sites on the protein that are most likely to bind an antibody. So for shorthand, we'll call those B-cell epitopes, although at this level it's a computational prediction. It's not a measurement. And the prediction that we and others have done is you make a purely biophysical calculation. This has nothing to do with immunology. It's just asking which features are easy for an antibody to grab hold of. And so we've come up with an algorithm that you can apply to the three-dimensional structure. And what's shown over in the upper right-hand corner there is take a sliding window across the sequence and make the calculation and find out where there's peaks that are the predicted B-cell epitopes. And then it turns out if you actually take those peptides and you immunize an animal with them, the antibodies that result with pretty high consistency end up binding to the protein. So these are reasonably good probes that we can then use to put on our different colored beads and in a parallel process pull out antibodies against this epitope, that epitope, and this epitope. Now, in order for this to work, the computational technology has to be pretty good. And until recently, the computational methods were not very good. So this is a review article from 2008, took all of the different B-cell epitope prediction algorithms that were available and applied them to a uniform data set that was 39 different epitopes where you actually know what the epitope is because it's an antibody antigen complex that's been crystallized, so you know exactly where the epitope recognition is. And most of the software was terrible, is that it predicted only 10% of the empirically defined epitopes. Some of them were up in the 40% range, and the technology that actually Trellis acquired by acquiring another company is actually up at about 85%. So that puts it in the range of accuracy of prediction that Tcell epitope in silico methods use. One of the nice features about the technology is that it's not restricted to finding linear epitopes. The calculation is done as a sliding linear window, but when you map those values back onto the three-dimensional structure, you can actually find ones that are discontinuous epitopes. So the blue, yellow, and red there are different linear regions of the EGF receptor that are recognized by Erbitux. And so even though they're disjointed in terms of the linear sequence and the three-dimensional, they're quite close to each other. So to start to summarize the talk then is the notion here is that if there's a structure available, we can use that to predict the B-cell epitopes. We'll use those as probes for the highly parallelized, multiplex, very sensitive human B-cell cloning technology. And there we would like to look at patients that are enrolled in the clinical trial. We can look at blood samples early, middle, and late in their exposure. And we can generate quite a lot of data on the frequency of ADAs for multiple epitopes. We can look at within a single patient evolution of the ADAs. We can look at patient population profiles. And that's just at the survey level. If we then go to the additional effort of actually cloning the anti-drug antibodies, now you can characterize those ADAs by epitope recognition, identify which of them are neutralizing antibodies using whatever assay of function is relevant. And of course, now you have reagents that you can construct competitive binding assays so that now serum becomes much more useful because you can deconvolute the components of serum by looking through different data channels defined by the competitive binding. And then on the flip side, once you have those monoclonal antibodies, then you can test them for human tissue binding, you can assess side effect profiles, you can correlate presence with clinical indicators. And then long term, if you have antibodies against the different epitopes and you find that one of them is particularly problematic, now you can make mutations in order to reduce the immunogenicity of that site. And that would then be a second generation product that would have reduced immunogenicity. Final slide is ADAs is potentially a killer for the biotech industry. Is that as an industry we got lucky that the anti-EPO response was so rare that it was really a vanishingly small number of patients, and it was an approved drug that was beneficial to so many people, and the FDA allowed it to stay on the market. If that had been 1% of patients instead of 0.01% of patients, and it had shown up during the clinical trial, it's very hard to see how the drug could ever have gotten approved. So going forward, the FDA is really nervous about all this because biologics are becoming the dominant, in 10, 15 years, they're going to be the dominant component of the pharmacopoeia. So we need, as an industry, we need a way to address the problem, and we have to do it in a way that's a reasonable amount of work. Is that we can't run clinical trials on hundreds of thousands of people to look for rare events. we have to have some more efficient way of dealing with the ADA problem. So from Trellis's point of view, this is not our main line of work, is that our main line of work is to develop therapeutic antibodies as drug candidates. But like most platform companies, you use the platform and you develop a candidate and then you spend a year characterizing it, and in the meantime your platform is sitting there idle. So we would like the platform to be used as much as possible, and that means that we would like to make it available in the ADA context just on a service model. Is that if we were to try and have any reach-through royalties on the drugs, that would be a non-starter, and we recognize that. So in terms of what's involved, is the survey is it's about 10 days per cycle, and probably you do it a few times and then you refine the reagents to get more information. And so, on the order of 3 months to do survey work on the order of 40 blood samples. Typically, we would like to get 50 cc's of blood. The partner needs to supply the drug substance, any QC reagents, a rabbit serum against the antigen, against the drug, or murine monoclonal, anything that when we put the drug onto our bead, we would like to verify that it still can be recognized by an antibody. And then we'll determine the ADA profiles across the individual patients. And then if it looks appropriate, then we can go back and we can actually clone the antibodies, generate a few milligrams of each, deliver that to the partner for toxicity assessment and off-target reactivity. So that's what we do, and that's what we think we can do that would be helpful to people interested in ADAs, and thank you for your attention.
Thank you, Larry. That's great. Any questions?
Have you had any interest or thoughts about developing this to look at isotypes or subtypes of antibodies?
You mean like IgG versus E and things like that? Yeah. So that's strictly whatever you coat the surface with. Is normally we coat with a GOAT anti-IgG, but we could easily do it for IgA or IgE. So yeah, that's completely straightforward.
So Larry, excuse my ignorance, but how practical is it to identify an epitope on a protein, then by making point mutations, remove that epitope? And will you not just create a new one?
Yeah. So it's a good question, and the best example to date is the work that Ira Paston has done on the Pseudomonas exotoxin PE38 that he conjugates to an antibody as the payload of an antibody targeting a tumor. So he makes the PE38 antibody conjugate, puts it in man, and gets great results. Tumor starts to regress. He gives a second dose and there's no effect. And the reason there's no effect is because the PE38 is immunogenic, and second time around the complex just gets cleared. So he started mutating PE38 in a very laborious way. He would make mutations all over it, inject them into mice, collect the monoclonals, measure them. About half a dozen years' work, probably half a dozen postdocs. And at the end of the day, he's got a PE38 that when he injects it into a mouse does not induce an immune response. It is immunologically silent. Unfortunately, when you take the blood from the patients that had received it, there are still some antibodies in that serum that are recognizing epitopes that were not recognized in the mouse. So if we could get permission to work with those blood samples, which are now owned by his pharmaceutical partner on those clinical trials, we'd like to be able to clone those antibodies out so that we can pinpoint where they're binding and then erase the immunogenicity. So the answer is, I believe it is possible, but it takes some work. No, it turns out that you don't.
With interference, we have 5 or 6 mutants
tested with point mutations and the bioactivity will always went.
Yeah. No, that's why I say it's not going to be an easy thing to do, but I think it's a feasible thing to do. So in the case of P38, it's about 8 or 10 amino acids that need to be altered, and it doesn't affect the biological activity as a toxin. So depending on what the protein is, factor 8 is going to be a lot harder. There's 35 different epitopes there, and they're intermingled amongst a bunch of critical regions and so forth. Yeah. One question that often comes up is, can we do the same thing with monkey blood? And we thought it would be easy until we tried it, but macaque B cells, if there are people who know how to grow macaque B cells better than we do, is that they're much more fragile, and we needed a different cytokine cocktail, and even so, we have much greater difficulty cloning antibodies from macaque. So, yeah.
Hello?
Yeah.
So earlier this year at EIP, there was a presentation from people at Sanquin about cloning, the different methodologies of cloning B cells for involved in the ADA response to Humira. And they found that, I guess, all of the people who developed ADAs developed neutralizing responses, and all the neutralizing B cells were against the same epitope. So, okay, the research didn't go as far as saying they've been able to remove the epitope, but at least, it's a single epitope region that they were looking at, and at least you know what you have to potentially work on. And it was in the CDR, so whether it's then feasible to make any changes
Right.
the antibody will still work is questionable, but at least in principle there's the possibility maybe to make a conservative substitution and achieve something.
Yeah, Humira, according to the computational technology I described here, it lights up like a Christmas tree right in the CDR3 region there. Yeah. Okay, thanks.
That's great. Thank you very much.